See that one arrow is lapping the other, so the frequency is higher in one than the other. And then you can just watch this little animation, and you can see if you just if you stare at it long enough, what's going on here. That if you look at the green and the blue, or purple rather, cell here, watch how they behave. They're out of phase now. They're oscillating out of phase, but then they come into phase. Now they're going together, right? You can see that now they're out of phase again. You can see the two oscillations moving in and out of phase with each other as you watch these two different theta cells that are bursting at different frequencies interact with each other. And you can imagine that if I have a grid cell over here that's listening to those two theta cells, and this cell fires only when those two cells are bursting together, but not when they're bursting separately, you can see how the grid cell will fire a lot when they are bursting together like now, but it will fire less as they move out of phase with each other, and then that will start to fire more as they fire together. And if this is all happening in the brain of a rat that's running down a track, the grid cell fires and then goes quiet, and then fires and then goes quiet at regularly spaced intervals along the track. So this is our sort of a model for how grid cells are formed from theta cells. Yes?
What frequency does the grid cell fire? Is there a way to figure out based on those?
Yeah, so that's a great question. At what frequency does the grid cell burst, I think you mean?
Yeah.
So if, let's say that as in the example, well let's change it. So let's say that one of these is, that the green is going at 8 hertz and the blue is going at 6 hertz, then what is the burst frequency of the good one? What is it?
2 hertz.
Not 2. Oh well. So wait. Well, there's 2 frequencies. Okay. So we have to. There's there's this frequency, like how often the grid cell fires, and then it goes quiet, and then it fires. We'll call that the beat frequency or the envelope frequency. But then when it fires, it's also going. It's bursting at what we call the carrier frequency. So there's actually 2 answers to this question, okay? So if the green is 8 and the blue is 6, what is the carrier frequency? 7, which is the average of 6 and 8. That's the bursting frequency. And what is the envelope frequency or the beat frequency? That's the difference, which is 2. That's right. So that part you will need to be responsible for and know. And that's in this slide here. You can see the calculation for that. So, other questions about that?
Can you go over that again? Yeah, what's the burst frequency and what's the envelope frequency?
So this grid cell, and again, okay, this is like this is we're doing our Fourier synthesis thing here as well. It's easier to show if we look at this, okay? So we have a 7 hertz oscillation and an 8 hertz oscillation that are being added together, and they give us this new waveform that looks like this. This is a signal that is produced by adding these 2 things together. And when you look at this signal, you can see 2 different oscillations superimposed on each other. There's this slow oscillation of the waxing and waning amplitude, and there's this faster oscillation of the bursting inside it. And when we think about the grid cell, the slow oscillation is the rate at which the rat is moving through the places where the grid cell likes to fire, which is slow, like once per second, maybe twice per second. But then there's this other frequency, the carrier frequency, which is the rate at which the cell is bursting as it moves through those areas, and that's a higher frequency component. And with these simple beat oscillations, the envelope is always equal to the difference between the 2 wave frequencies that are added together. And the carrier is the average when we're dealing with 2. Yes?
Which frequency is the one shown on the power spectrum?
What's that?
Which is the one that's shown on the power spectrum?
Which is one which is shown on the power spectrum of this. If you take a power spectrum of this.
Is it like when they have just like the one like high?
Well, so we've looked at if we took the power spectrum of that right there, then it's a good question. What would it look like? It would have two bumps at what frequencies? 7 and what? 7 and 8. Right. Did anybody want to say 7.5 and 1? Did somebody say? Did you? Okay. Because I mean, it's a sensible answer. Okay. I mean, it's wrong, but I can see why you would say that. But let me say why it's wrong. It's kind of important because by definition, the power spectrum is telling you what frequencies need to be added together to get the wave that you have, and these two waves added together give you this. You actually can get this by combining a 1 hertz and a 7.5 hertz wave, but you multiply them instead of adding them. And the Fourier transform does not tell you what you get by multiplying, it tells you what you get by adding. Yes?
So each envelope is a dot on the grid cell, or
Each of these is yeah, you can think of this as the rat passing through one of the spots where the grid cell likes to fire. It's going into the grid, into that little field, and then out again, and then it's going to the next one and out again. So if we look at a grid cell, here's a grid cell. It's like he's running into this and then out, and then into this and then out. Okay. Yes.
So in the actual data we looked at, where like theta cells show frequency or burst frequency around 7, right?
Mm-hmm.
Does that mean all of them have a frequency of 7?
The grid cells?
All the theta cells that combine to in the actual data we looked at during lab.
Well, so according to this, like when the rat is running at whatever speed this is, you have sort of two populations of theta cells here, the green ones and the purple ones,
Yeah.
and they're going at different frequencies.
Yeah.
So not all the theta cells should be going at the same frequency at any given time. And that's actually important.
Right.
That's one of the things that we said has to be true for this mechanism to work, and that's why we talked in the third lab about phase precession and how that shows us that there actually are more than one theta frequency. There's not just one theta frequency, and that's actually critically important.
Why for the oscillator for the grid cells do you have 2 arrows when you were doing the previous example was of a theta cell, right? The previous oscillator was of a theta cell, right?
You mean this?
Not this one. The previous oscillator
Yeah.
was of a theta cell, right?
This.
Yeah. Okay. Yeah.
It's
that
of
makes
many
sense.
theta
Yeah.
cells. It's not just one. Every cell in that ring is a theta cell.
Okay.
And they're all bursting at the same frequency, basically, but at different phases. And this is also many theta cells, but we have 2 separate populations. One group that's bursting at a slow frequency and one at a faster.
so with the place cell, you just have a lot of theta cells on top of each other, the same.
Yeah, to get a place cell, you need not just 2 rings, but 8 of them or something. That's right. Yes.
So you said that like the rats obviously don't just like spin in circles regularly.
Right.
So how do we get like regular, and they're probably not going to move through their preferred spots. How do we get like regular wave patterns
For this?
actual movement?
You mean for this? So, you mean why did these spin?
I guess like if we look at this graph, it like goes regularly up and down. Like we're not actually going to see that in real data, will we?
Well, that's what a grid cell does. You mean this thing here, right?
Yeah.
So you can think of that. So here you have a rat that's running across the floor, and as it does so, this grid cell fires at this spot on the floor, and then not at this spot on the floor, and then at this spot on the floor, but not at this spot on the floor, right? And that actually is, in real data from grid cell recordings, what we do in fact see. So here's a recording of a real grid cell, and it fires at these spots here. So if the rat was, say, running along this line, the grid cell would fire at this spot, and then not at this spot, and then at this spot, but not at this spot. So we actually do see, that's one of the things that makes grid cells sort of so remarkable, is they do, in fact, draw this spatial oscillation on the floor. That's what they actually do.
But when like the undulations aren't gonna be regular, right? So I'm looking at like for Fourier.
Oh, you mean like
Fourier
the,
analysis, like
okay,
you're trying to break it
yeah,
down.
that's right. So I think what you're getting at is the thing that we didn't have time to go into, which is that these oscillations are fixed in space but not in time, and the Fourier spectrum gives you this that's right, because if the rat runs quickly through here, if the rat runs slowly, you'll hear but if it runs quickly, you'll hear so the frequency will be different depending on its running speed. We didn't quite get into how that gets all resolved, but you're right. So it's not going to be the same tempo frequency in both cases. But don't worry about that. The main thing is to grasp that the rate at which the rat moves in and out of the bumps is much slower than the bursting frequency, and so you end up with these slow envelope and this fast carrier component. That's at a fixed running speed. Don't worry about varying. Questions? Yes.
So at like a fixed running speed, are you saying like there's different theta cells that are gonna respond differently, like they have different frequencies, and that's what causes them to match up like this?
Yes, that's exactly right. At a fixed running speed, there's different theta frequencies, and that's what the phase precession that we talked about this week is showing us, that there actually are different theta.
There's different groups of theta cells that respond differently to the same speed.
Yes, they fire at different rates during the same speed. That's what we like to think. And actually, definitive proof that individual theta cells do this in the way they should is still not, does not exist yet. That's actually what we're trying to do in our lab right now. But it is unquestionable. The phase precession data that I showed in lab this week, which we will get to, shows that you can measure different frequencies of theta rhythms simultaneously in a behaving rat. So that's a good thing, basically.
So it's like, it's not like an individual cell, it's like a group of theta cells that like for a given speed like respond at 7 hertz.
That's right. Well, in this example that we're looking at, you have two groups of theta cells, the purple and the green, and at any given running speed, the purple cells will always be going at the same frequency as each other,
Yeah.
but at a
But
different
different
frequency
than
from the
the
greens.
greens.
That's right. That's exactly right. Other questions? Okay. So, this is just showing us that this theory of how grid cells are formed, which is illustrated in that diagram, it requires grid cells to get input from theta cells, and they in fact do, and this is this lovely result from the Brandon paper where if they, Brandon and Koenig, I should say, they both did this experiment and published them back to back in Science, that if you turn off the medial septum with a drug, grid cells basically stop working. So that's shown in this diagram, that if you record a grid cell, so here's a grid cell, and then you turn off the medial septum by putting a drug in there so that the theta rhythm basically, the theta rhythm inputs to the grid cell are messed up. Then the grid cell gets messed up. You see the grid is gone here. But if you wait 24 hours for the drug to wear off and record again, the grid cell comes back. And that's just shown twice in two different cases here for different grid cells. So does that make sense? It's a pretty simple result. Yes?
In the pathway, it can also go through the anterior.
Yeah, that's right. Mm-hmm.
So then, wouldn't you still see firing?
Well, that's a good question. Why don't you? And nobody can say that for sure. It's a very good question. But it becomes perhaps even more relevant when we look at what happens to place cells when we do the same manipulation. And they actually don't go away. They don't stop firing. And what you just said might be a possible explanation for that. Again, nobody knows for sure that it is the explanation, but maybe
So
place cells
in
are
that
using
case,
that data.
That secondary projection is just a kind of place cells.
Yeah, so maybe, and this is just speculation because more studies need to be done, but maybe the medial septum projections are more important for the grid cells to do their thing, and the thalamic projections are more important for the place cells to do their thing. Who knows? Yeah. Yes and no. So there is a study where they did that, and basically, this was before grid cells had even been discovered, and they did this to